Welcome to the SWAYAM NPTEL course on food microbiology for safe and sustainable food systems. In the last lectures, we were discussing microbial growth and we have seen the various factors affecting the growth. Now, in this lecture, we will be measuring the bacterial growth. And the things we will include, that is the direct method, dry wet method, wet weight method, chemical measurement of cellular components, and indirect method like role of composition of metabolic products, photoelectric study, that is nephelometer, measurement of cell number, that is the total count, and total count we will be measuring by electronic counting chamber and direct microscopic count, and viable count by pore plate, spread plate, Miles and Mishra method, membrane filtration technique, and most probable number, that is MPN. Now, measurement of bacterial growth. There are many ways to measure microbial growth to determine growth rates and generation times. Either population number or mass may be followed because growth leads to increase in both. Here, the most commonly employed techniques for determining population size are examined briefly, and the advantages and disadvantages of each will be discussed. No single technique is always best. The most appropriate approach depends on the experimental situation. This figure represents the different methods of measurement of cell mass. There are two methods, that is direct method and indirect method. The direct method includes physical method and chemical method. The physical method includes dry wet basis, wet basis, and the chemical method includes the chemical measurement of cellular components, whereas the indirect method that includes chemical activity method and the turbidity measurement. Chemical activity method that includes the rate of composition of metabolite, rate of composition of metabolic product, and turbidity measurement that includes the photometric study. Now, the direct methods. Direct physical measurement of dry weight, wet waste, or volume of cells after centrifugation. Direct chemical measurement of some chemical component of the cells such as total nitrogen, total protein, or total DNA count. Indirect methods. The indirect measurement of chemical activity such as rate of oxygen production or consumption, carbon dioxide production or consumption, etc. Measurement of cell mass by turbidity. The turbidity or optical density of a suspension of cells is directly related to cell mass or cell numbers. The method is simple and non-destructive, but the sensitivity is limited. Turbidity is limited to relatively high cell concentrations, approximately 10 to the power 7 cells per ml for most bacteria. Dry weight method. The most direct approach is the determination of microbial dry weight. Cell growing in liquid medium are collected by centrifugation, washed, dried in an oven, and weighed. This is an especially useful technique for measuring fungal growth. It is time consuming, however, and not very sensitive, because weight of bacteria is so little, it may necessary to centrifuge several hundred millimeters of culture to collect a sufficient quantity, that is milligrams dry weight per ml. Advantages. only one way to determine growth of filamentous bacteria. It is rapid and easy, but disadvantages are it is cumbersome, not very accurate, it does not give cell numbers or increase in mass. It cannot distinguish between live and dead cells and must work within certain absorbency. Now, wet weight basis, same as dry weight method, but the wet weight of cells taken into consideration. Now, the chemical measurement of cellular component, that is total nitrogen content. Major constituent of cell material is protein, and since nitrogen is a characteristic part of protein, bacterial population can be measured in terms of bacterial nitrogen. On an average, bacteria contains approximately 14% nitrogen on a dry weight basis. To measure the growth by this technique, first harvest the cells and wash them free of medium and perform a quantitative chemical analysis of nitrogen. Limitations: it can be performed only on samples free from all other sources of nitrogen. It is applicable for concentrated solutions only. Now, the rate of consumption of metabolic products. An indirect way of estimating bacterial numbers is measuring the metabolic activity of the population. For example, acid production or oxygen consumption, nutrient utilization, waste production, pH, etc. The assumption is that the amount of acid produced or oxygen consumed under specific conditions and during a fixed period of time is proportional to the magnitude of bacterial population. Admittedly, the measurement of acid or any other end product is a very indirect approach to the measurement of growth and is applicable only in special circumstances. Turbidity measurement or optical measurements. A quick and efficient method of estimating the number of bacteria in a liquid medium is to measure the turbidity or cloudiness of a culture and translate this measurement into cell numbers. This method of enumeration is fast and usually preferred when a large number of cultures are to be counted. The technique is based on the fact that microbial cells scatter light proportionally within certain limits of their concentration. A spectrophotometer is used to determine turbidity, that is cloudiness, by measuring the amount of light that passes through a suspension of cells. When a beam of light is passed through a suspension of bacteria, there is a reduction in the amount of light transmitted as a consequence of scattering, which is a measure of cell density. This is done by spectrophotometer, which measures in the absorbency unit, that is A. Absorbency is determined as the log of the ratio of intensity of light striking the suspension, that is I0, to the transmitted by the suspension, that is I. That is A is equal to log I0 by I. This measurement is very convenient for measuring the density, provided it is calibrated against bacterial suspension of known density. More cells is equal to more turbidity, More turbidity is equal to less light passing through the suspension. The lower limit of this method is reached when the population contains bacteria about 10 millions per ml. Since the scattering S is inversely proportional to the fourth power of the wavelength lambda, that is S is equal to 1 by lambda to the power 4 of the light being used. So, sensitivity of the measurement increases sharply if light of shorter wavelength is used. More sensitive instrument is nephelometer. These have the light sensing device arranged at right angle to the incident beam of the light and hence directly measure the scattered light. Nephelometer. Nephelometry, from the Greek word nephelo, which means cloud, is an analytical chemistry technique used to measure the amount of turbidity or cloudiness in a solution caused by the presence of suspended insoluble particles. When directed through a turbid solution containing suspended solid particles, light is transmitted, absorbed, that is blocked, and scattered, that is reflected off the particles. The amount of scattered light depends on the size, shape and concentration of the insoluble particles in the solution, as well as on the incident wavelength of light. A nephelometer is a dedicated standalone instrument that measures the intensity of scattered light. Transmitted light is not detected. The basic components of a nephelometer include a light source, light scattering optics and a detector. The light source generates a beam that is directed through the sample. Halogen and xenon lamps or lasers can be used as light sources. Lasers are typically the most common choice due to their sensitivity, high intensity and coherent nature, that is emitted photons are in step with each other. Incoming and outgoing wavelengths are identical and hence not optically selected. A detector is placed opposite the light source and at an angle relative to the incoming light beam. It detects variations in either forward angled scatter or side scatter depending on its position. Depending on the angle at which the most scattering can be collected, detectors may be placed at angles of 30 degree, 70 degree or 90 degree. This figure shows the difference between a turbidity metric and the nephelometric detection. This figure A shows the light source which directly falls on the suspension which is containing the microorganisms or the suspended solids, and transmitted light is measured. Here are the detector which measures the transmitted light only, but in the nephelometer it detects the scattered light. Here is the detector, it is placed at a 90 degree angle. The measurement of cell number. The cell number can be measured either by total count or by viable count. The total count includes the electronic counting chamber and direct microscopic count, whereas the viable count includes plate count, membrane filtration technique or most probable number. The plate count is of three types: one is pore plate count, another is spread plate count and another is Miles and Mishra method. The total count. A total count is a direct counting method in which all cells are counted, whether dead or alive. Requires the employment of microscopes. Now, the viable count. A viable count is a direct counting method in which only live cells are counted. Viable counts can be accomplished by techniques such as pour plating, spread plating and most probable number method. Now, electronic counter chamber or Coulter counter. Larger microorganisms such as protists and yeasts can be directly counted with electronic counters such as the Coulter counter. Although the flow cytometer is increasingly used. In the Coulter counter, the microbial suspension is forced through a small hole or orifice. Electrical current flows through the hole. The electrodes placed on both sides of the hole measure electrical resistance. Every time a microbial cell passes through the hole, electrical resistance increases, that is, the conductivity drops and the cell is counted. Each time a bacterium passes through the orifice, the conductivity drops, which is recorded electronically. The instrument can score the magnitude and duration of changes in the conductivity and register or record the number and distribution of size of a culture population respectively. The orifices commonly used to count bacteria are 30 micrometer in diameter. The medium must be strictly free from dust, etc., because lower dust particles will be recorded as bacteria and higher will clog the orifice. Flow cytometry gives accurate results with larger cells and is extremely used in hospital laboratories to count red and white blood cells. It is not as useful in counting bacteria because interference by small debris particles, the formation of filaments and other problems. The number of bacteria in aquatic samples is frequently determined from direct counts after the bacteria have been trapped on membrane filters. This is the pictorial representation of the Coulter counter. The bacterial suspension is passed through an orifice which is connected with two electrodes, and each time bacteria is passing through the electrode, the conductivity drops and which is recorded, and the time for which the conductivity drops, that will approximately give the size of the bacteria. The direct microscopic count. The direct microscopic count is a determination of the number of microorganisms found within a demarcated region of a slide known to hold a certain volume of culture. Use special slides known as counting chambers, for example, Petroff-Hauser counting chamber, Neuber chamber or Helber counting chamber. Another method is Breed's smear technique. This Petroff-Hauser counting chamber consists of this A, that is the side view of the chamber showing cover glass and the space beneath it that holds the bacterial suspension. This groups that holds the bacterial suspension, which is covered with the cover slide, and this is the top view of the chamber. The grid is located in the center of the slide, and this is the whole view of the grid where the bacteria in several of the center squares are counted. Usually at 400 to 500 magnification is used. The average number of bacteria in these squares is used to calculate the concentration of cells in original sample. Since there are 25 squares covering an area of 1 millimeter square, the total number of bacteria in 1 millimeter square of the chamber is number of bacteria per square into 25 squares. The chamber is 0.02 millimeter deep, and therefore, bacteria per millimeter cube will be equal to bacteria per square into 25 squares into 50. The number of bacteria per centimeter cube is 10 to the power 3 times of this value. So, for example, suppose the average count per square is 28 bacteria, then bacteria per centimeter cube will be 28 into 25 into 50 into 10 to the power 3, that is equal to 3.5 into 10 to the power 7. The Breed's smear technique. This technique is used commonly for counting the bacteria in milk. A grease-free slide is placed over a template of 1 centimeter into 1 centimeter, and a 0.01 ml of sample is spread over this area. The smear is allowed to dry, fixed by heat, and stained with methylene blue for 1 minute. After drying, the smear is examined under oil immersion objective and the bacterial cells are counted. So, bacteria per ml in sample will be equal to N into area of smear divided by area of one field into 100, where N is average number of bacteria per field, R is the radius of oil immersion field, that is 0.08 millimeter, and area of smear is 100 millimeter square. Limitations. Dead cells cannot be distinguished from living ones. Only relatively high concentrations of bacteria may be enumerated, that is more than 10 to the power 7 cells per ml. Dilute samples have to be concentrated by centrifugation or filtration. Now, the plate counts. The plate counts involve spreading a sample of a culture on a nutrient agar surface after diluting the sample or cell suspension in a non-toxic diluent, that is water or saline. If plated on a suitable medium, each viable unit grows and forms a colony. Each colony that can be counted is called a colony forming unit, that is CFU, and the number of CFU is related to the number of viable bacteria in the sample. Advantages. Sensitivity. Theoretically, a single cell can be detected. Always allows for inspection and presumptive identification of the organism on the basis of colonial morphology. Limitations. Only living cells develop colonies. Cell clumping can lead to an undercount of viable cells. Colonies develop only of those organisms for which the culture conditions are suitable for growth. Require lengthy incubation period, about 24 hours, for colonies to become visible. Prevention of crowding often requires serial dilution. Too few cells require concentration, for example, by centrifugation or filtration. Typically, a plate with CFUs between 30 and 300 is selected for the purpose of counting. Now, the pore plate. The pore plate method is employed for viable cell counts, isolation and pure culture. 10-fold serial dilutions of the test sample or suspension of microorganisms are prepared in molten agar at a temperature 45 to 50 degrees Celsius. The molten medium is then poured onto Petri plate or Petri dish and allowed to solidify, each dilution in a separate plate. Colonies appear within, beneath and on top of the agar after incubation. Disadvantages: the cells do not develop typical colonial morphology and are not easily accessible for future testing. It is not useful for temperature sensitive bacteria. It is difficult to count subsurface colonies. This is the pictorial representation of pore plate method. Here the inoculum is spread on the Petri plate, then the molten nutrient agar at the temperature 45 to 50 degrees Celsius is poured, then the whole mixture is mixed homogeneously and then it is incubated. The colonies grow on agar surface and subsurfaces. Now, spread plate. The spread plate method is employed for viable cell count, isolation and pure culture. Prior to inoculation, 10-fold serial dilutions of the test sample or suspension of microorganisms are prepared in a suitable diluent. A small volume, approximately 0.1 ml of each dilution is then inoculated onto the surface of agar that has already solidified in a Petri plate and then spread around the agar surface. The plates are incubated for 24 to 48 hours at 25 to 37 degrees Celsius. Colonies grow solely on the surface of the agar. This technique is advantageous particularly when cells are sensitive to relatively high temperatures. This is the pictorial representation of spread plate technique, where the samples or the inoculum is poured on the pre-solidified agar medium, then it is spread and then it is incubated. In this case, the colonies grow only on the surface. Now, the Miles and Mishra techniques. This is a modification of spread method. This is a modification of spread method in which the agar plate is divided into 6 or 8 sectors. An inoculum of 0.02 ml is dropped on the surface of agar. The drops are allowed to dry and the plates are incubated for 24 to 48 hours at 25 to 37 degrees Celsius. After incubation, plate inoculated with a sample dilution yielding about 30 colonies is selected for counting using a colony counter. This is the pictorial representation where the sample or the bacterial suspension is diluted in 0.5 McFarland bacterial broth, then it is serially diluted 6 times each hour and then 0.02 ml or the 20 microliter solution is poured on the 6 chambers. This is dilution 1, dilution 3, 2, 3, 4, 5, 6. In the 6 they are just dropped on that and then it is incubated for 24 to 48 hours at 25 to 37 degrees Celsius temperature. The membrane filtration technique. In filtration, Bacteria are retained on the surface of a membrane filter of pore size 0.22 micrometer and then transferred to a culture medium to grow and subsequently be counted. If too few colonies are present, then the original culture must be concentrated prior to determining its plate count. This is a useful method for determining the number of bacteria in a water sample or other clear fluid where the bacterial number is low. This is the pictorial representation where the bacterial suspension is filtered through a membrane using the vacuum pump or vacuum device. The material which is retained on the membrane, that is either gone for the morphological analysis or it is serially diluted and then plate count for the bacterial cultivation. The filtrate also is subjected to bacterial cultivation. Now, the most probable number are MPN. The most probable number is a method used to estimate the concentration of viable microorganisms in a sample using broth of different dilutions. It is commonly used in estimating microbial populations in soils, waters, agricultural products, and is particularly useful with samples that contain particulate material that interferes with plate count enumeration methods. MPN is most commonly applied for quality testing of water. A group of bacteria, commonly referred as fecal coliforms, act as an indicator for fecal contamination of water. Water to be tested is diluted serially and inoculated in lactose broth. Coliforms, if present in water, utilize the lactose present in the medium to produce acid and gas. The presence of acid is indicated by color change of the medium, and the presence of gas is detected as gas bubbles collected in the inverted Durham tubes present in the medium. This is the pictorial representation of water testing, that water sample is diluted serially, that is 10 ml, or directly it can be inoculated in the lactose broth of 10 ml of lactose broth, and Durham tubes are inserted here. 10 ml, 1 ml, 0.1 ml can be inoculated. Then after incubation, if the color is changed, here we can see the 4 positive tubes where the color is changed due to production of acid, and gas is also produced in the Durham tube. Here you can say that only 2 test tubes are positive, and here none of the tubes is positive, means all negative test. That is all for today. Thank you very much.